So good afternoon. We are proceeding with the fifth session, the title Phage Therapy, Belgium Perspective. I would like to introduce the speaker, Dr. Jean-Paul PIRNAY. Sir is the head of the Laboratory for Molecular and Cellular Technology, Queen Astrid Military Hospital, Brussels, Belgium. He is involved in the study of molecular epidemiology, including antibiotic resistance or bacterial pathogens, and the development of therapeutic phage preparations. The moderator would be Dr. Hiran Joshi, and the panelist myself. Sir, it's all yours, sir.
Good afternoon. Let me start by thanking the organizers for the invitation to present our research. So I will talk about the phage therapy system we developed in Belgium. But first I will have a few words about the military hospital where I work at for about 30 years now. So it's known for its referral burn wound center, but it's also what one calls a calamity hospital. And this means that it's equipped and trained to respond to mass casualty incidents. An example, the 2016 Brussels bombings, there were 32 deaths, more than 300 injured, and most of them passed through the military hospital. I will show some, one slide actually that contains some potentially sensitive material. Here it is. So these are the wounds we typically saw then, very comparable to battlefield wounds, burns, open fractures, penetrating injuries. And it's not hard to imagine that these wounds are prone to bacterial infection. And as you all know, AMR is becoming a big problem with an estimated 8 million deaths associated with AMR in 2050. So that's why we decided to research phage therapy in the military hospital about 20 years ago now. You all know what phage therapy is, so I don't have to explain. You can see a nice picture, helium ion microscopy picture of phages attacking a bacterium, a few bacteria. Now, in these 20 years, we have developed an entire chain of phage therapy research in the military hospital, and we go from isolation of phages in the environment on the left to the application of phages on the right. Of course, in between, you have a lot of research and production. And we collaborate with a lot of external institutes, but the most important one is the Eliava Institute in Tbilisi, in Georgia. And as you know, they have more than 1 century of phage therapy experience. Now, phages are a medicine, but they are quite different from conventional medicines such as antibiotics or aspirin. I will just discuss two of these peculiarities. One is selectivity. So as you know, phages only target a subset of one bacterial species. Antibiotics, when they still worked, they used to target a lot of species, for instance, all gram-negatives. But phages, they only target a part of one bacterial species. Now, this has an advantage because you can choose phages to only target the bacterial pathogens and to spare the beneficial commensals in your intestines, on your skin, for instance. But it has also a huge disadvantage. So I think this is the main disadvantage of phage therapy. That is that we need a handful to dozens of phages to target only one species. And now, why is that a problem? Well, when we analyze the phage therapy requests in Belgium. You can see in Brussels on the right. I also did this in San Diego on the left. You can see that we need phages targeting more than 30 bacterial species. You can also see that these pie charts are not identical, so there are geographical variations. And I can assure you that there are also variations over time, not only in the species that will be more prevalent, but also in the strains within these species. So this means that we need hundreds of phages and that they also need to evolve with the bacterial pathogens. And this is a huge problem. Then there is resistance. So bacteria also become resistant to phage. And you all know about CRISPR-Cas. This is a known antibacterial defense, anti-phage defense mechanism of bacteria. But as you can see in the image to the left, I think almost every month one discovers a new anti-phage defense system in bacteria. And what we don't know is what will be the impact of using the same phage product over and over again at the same place on the selection of these systems, their persistence, and maybe also their spread in the environment. We have no idea. Still, with all these differences of phage compared to antibiotics, the regulator has decided that we need to follow the conventional drug development and marketing pathways for phage. And every phage product is considered to be another drug. So this means that each phage product needs to be manufactured according to good manufacturing practices. Each product needs to undergo preclinical studies, animal studies, toxicology. And you have to do this for every product. And then you need to perform the phase 1, 2, and 3 clinical trials, or so-called randomized controlled trials. And then you can apply for marketing authorization. And as I said, you need to do this for every phage product. This requires a lot of time and money, and someone calculated that on average it takes 10.5 years for a drug to get to phase 1, from phase 1 to regulatory approval. And the average cost of creating a new drug ranges as high as $2.6 billion. And again, you need to do this for every phage product. Now, some companies are trying to do so, but after what has been coined 2 decades of phage therapy renaissance, there are no drugs on the markets in the European Union and in the US. I don't know about India. There might be maybe some products on the market. I'm not sure. This is an interesting paper in Nature Biotechnology. It's a paper from 2004, which is about 20 years old. And it describes the budding phage therapy business. And in this paper, there's an interesting table, and it enumerates the phage therapy companies that were on the market, not on the market, that were trying to go on the market in 2004. And you can see, I think there are 12 or 13 of them. One is the Phage Therapy Center in Tbilisi, Georgia. I would not call that a company. And then you have, I think, 11 real companies. All of them founded between 1994 and 2003. 2 are still existing. there is Gangagen in India, and Intralytics in the USA. And interestingly, they both focused on agricultural and environmental applications. When we look at the situation today, we can find 25 clinical trials registered in several databases. Seven of them are completed without really good results, one can say. 3 terminated by the company, and 15 are still active or recruiting. 24 are in phase 1 or 2. There's one phase 2-3, but it was unsuccessful. Now, interestingly, when we look at the bacterial species they target, we find that they only target 9 bacterial species, and 80% of them only target 4 bacterial species. And as you remember, we need phages that target more than 30 bacterial species. In addition, when you look at the indications they target, it's also only a limited number of indications, and they go for indications with large numbers of patients, which makes sense because they want to have a big market. For instance, diabetic foot infections. So this means that even if all these products would come to the market tomorrow, but they are still very far from the market because most of them are in phase 1 or 2, it would not really solve our problem because these products will only target a handful of bacterial species and indication. And as you know, we need phages today because patients are dying of AMR today. So that's why in Belgium, we decided to develop a more pragmatic phage therapy framework. And we called it the Belgian Magistral Phage Medicine Concept. We have a phage bank with characterized phages. These are stored using a tiered banking system. From this, we take phage that will be used as a seeding lot for the production of a phage API. And API stands for active pharmaceutical ingredient. So we are making phage ingredients, and we need to produce them according to a monograph, which is some kind of a recipe. It's only a few pages large, which will describe how to produce the phage. And then there needs to be external quality testing, again, based on this monograph, because this monograph will also describe which tests need to be performed. And then these phages need to be approved by a Belgian approved laboratory. And then these phages can go to hospital pharmacies, where upon prescription by physician, these phages, so one or more phages, can be integrated into a magistral preparation. And this is a preparation made by a pharmacist. So at that point, several phages, for instance, against Pseudomonas aeruginosa, can be mixed in an hydrogel for a patient in the burn wound center, for instance. And I need to stress that these phages are not GMP certified. So this monograph that describes how to produce the phages will not ask for GMP certification. And there's also no marketing authorization needed. This doesn't mean that there is no quality. So we also indeed work with standard operating procedures. We work in a controlled environment. And on the bottom right, you can see two examples of phage magistral preparations. Important is the quality testing. And in Belgium, this needs to be performed by a Belgian-approved laboratory. And Cienzano is one of these laboratories. That sounds like a private company, but it's just a fancy new name for what was used to be called the Scientific Institute of Public Health. So this institute releases drugs for use in Belgium. For instance, they also tested and approved the COVID-19 vaccine in 2020, if I'm not mistaken. You go to the site of the Public Health Institute, and there you can see therapeutic bacteriophage preparations, safety of therapeutic bacteriophage preparations. And when we click on this, you can see what this means. So it means that these phages need to undergo quality control on two levels. So first, there is a genetic control, a check of the safety of a phage. And then there is a control of various parameters for each phage API production. Now, what does this mean? So the first, the genetic control means that we need a phage genetic passport. So this institute will analyze the genome and look and make sure that it's strictly lytic, the phage, that there are no known toxin or antibiotic resistance genetic determinants, and they will also screen the production host. So this is the bacterium in which we will produce the phage. Then for every API batch, they will check for the phage identity. So is this the phage we say it is? They will look at the phage titer, the pH, the bioburden, and the endotoxin level. And we are now together with this institute making some fine-tuning. We will probably this year add an overall pyrogenicity test that will likely replace the endotoxin test. Then we will look also for prophage threshold. We are allowed to have prophages in our productions, but the current monograph does not mention any threshold. And I think here we will mention one, which will probably be like around 5%. But most importantly, it's not the amount of prophage that is important, it is what's on the prophage. For instance, if this prophage would code for a toxin, we should not have it in our preparation. So then the threshold would be 0%. And then also we are allowed to pre-adapt the phage. Some people will say train the phage to make it better. Also there, the current monograph does not mention any threshold. So how far can you train to still have the same phage? So also there will be some percentages that we can deviate from the original phage. So since we have this system, our phages have been applied in about 200 patients now. And of course, we are not a phage company. We are a research laboratory in a hospital. So we cannot cater for all the requests. And we get like about one request per day on average. So we cannot deal with this. So we need to select the patients that would benefit most from phage therapy. And you can see on the right, we have created this multidisciplinary patient selection process. I will not go into details, and it has been published, I think in June last year. We decided to analyze the first 100 consecutive personalized phage therapy cases. This was a retrospective analysis, and study protocol was registered on clinicaltrials.gov. But we also published the results last year. It was very interesting because there was also an editorial by Nature Microbiology on this paper. And the editor mentioned, this is the first time I saw something like that. The editor wrote, the development and application of phage therapy represents not only a promising, but also a mandatory alternative approach to tackle this major public health threat. And this public health threat is, of course, AMR. But it's the first time that I see someone really writing that it's a mandatory alternative. In these 100 patients, we targeted a lot of different indications, different infection types. So we did not focus only on indications with a lot of patients because we have no commercial incentive. Our incentive is to treat all patients with difficult to treat bacterial infections. So most of them lower respiratory tract infections, and then some skin and soft tissue infections, bone infections, and then upper respiratory tract infections, bloodstream infections, and others. This figure to show that we also treated 5 children that were younger than 1 year. You can see that in these first 100 patients, we targeted 14 bacterial species. And as I said, all companies together, they only target 9. So here in these first 100 patients, we target 14, and sometimes 3 species in 1 patient. Now for this, we needed combinations of 26 phages, and 13 had to be pre-adapted or trained. And they were often used in combination with antibiotics. Now, I had a quick look at what this would mean for the first 200 patients. Well, this means more than 80 batches of more than 40 phages. Now, imagine doing this in a conventional system. So producing 80 batches of 40 phages according to GMP, then doing preclinical trials and randomized control trials. That would be impossible. Or even if it would be possible, it would cost so much money and also so much time that it would not be feasible. Some results. We saw clinical improvement in 77% of cases, eradication of the targeted bacteria in 61% of cases, and we saw 70% less probability of eradication without antibiotics. So this sentence says, try to use phages together with antibiotics. You will have more success. Some other interesting findings. Well, we saw selection for bacterial phage resistance in 7 out of 16 patients for which we had sufficient samples to look for this. So you need samples before, during treatment to analyze the selection of bacterial phage resistance. And I will not discuss this because Sabrina already did this an hour ago, I think. But I think I'll just show you this again because I think what is important is that we showed for the first time that even using a cocktail with 3 phages targeting 3 different receptors is not enough to prevent phage resistance. So this patient 91, was treated with 3 different phages, PT7, PNM, and 14-1. And as Sabrina explained, we got phage-resistant bacterial mutants that had mutations in all 3 receptors for these phages. And most of the time we see that these mutations are in the receptors. So you remember I mentioned these anti-phage defense systems like CRISPR-Cas. Well, in practice during therapy, we do not see these emerge. But of course, they play a very important role when looking for a phage to start the therapy with. But once you have a phage that is not targeted by anti-phage defense systems in the bacteria, then mutations in receptors become very important. But as Sabrina explained, we can try to live with these resistance because we can choose phages that then will cause, for instance, the resensitization to antibiotics, or will have a reduced virulence. Another interesting finding is that we could see a lot of in vitro phage antibiotic synergy. I will illustrate this. So in red, you see the growth curve of the patient's bacterium without phage, without antibiotics. You see the classical growth curve. Now, when we add the antibiotic aztreonam at 8 milligrams per liter, which is what you would find in the blood according to the manufacturer of this antibiotic, then you see an inhibition of the bacterial growth in yellow. So the yellow curve for, let's say, about 24 hours. But then you see regrowth. So these are resistant mutants. And you can also see that the curve will not go as high as the growth curve of the bacterium without antibiotic, because there's some kind of a fitness cost to this resistance. And then you can see in green the growth curve when we apply phage PNM at an MOI of 1. MOI stands for multiplicity of infection. It means that we apply 1 phage for 1 bacterium, which is realistic in therapy. And again, we can see that there is inhibition of the bacterial growth a bit longer, let's say 28 hours, but then again, regrowth. And again, you can see a fitness cost. What is really interesting is when we apply phage and antibiotics together, and again, aztreonam at 8 milligrams per liter and PNM at MOI 1 altogether. And then we see the blue growth curve, total inhibition of growth for 72 hours. Now, this patient was treated with a combination of phage and 3 antibiotics. So aztreonam, colistin, and gentamicin. And we see the same synergy of the phage with the 3 antibiotics. But it's not always the case. I think also Sabrina explained this, that this phage antibiotic synergy depends on the phage, on the antibiotic, and on the bacterium. So this is an example of colistin and phage M1, and you see no synergy at all. Then we also saw phage immune neutralization. So this is neutralization of the phage activity by the patient's immune system. And we saw this in 38.5% of screened patients. So again, it's only in 13 patients, although Now today we have more data, but in 100 patients, we had 13 for which we had sufficient serum samples before, during, and after phage therapy to try to document the phage immune neutralization. And in 5 out of 13, we saw this neutralization emerge. It emerged after 6 to 35 days after initiation of invasive phage therapy. I put invasive in bold because we did not see it, for instance, in topical phage therapy. So this invasive is mostly intravenous phage application. And again, it depends on the phage and on the patient. Then I explained that we had to pre-adapt or train the phage in 13 of the 100 patients. Now, we know that when we pre-adapt bacteriophage, we can get increased pathogen clearance and lowered resistance evolution. This means, it doesn't mean that there will be no bacterial phage resistance, but we know that it will occur later. When we do tests in vitro, for instance, and we see bacterial phage resistance emerge after a few hours, then we feel that this phage is not a good candidate for phage therapy. And then we will train the phage, and then often we can see that this resistance will emerge after, for instance, 48 hours, which is suitable for phage therapy. When we look at what this training or pre-adaptation means on the genomic level, then we see again that most of these mutations will be in the tail fiber of the phage. So where the bacterium escapes phage predation through mutations of the receptor, a phage can increase its activity by mutation in the part of the tail fiber that connects to the receptor. But sometimes it's even not really in the part that connects to the receptor. It can be, for instance, higher. And in this case, for instance, you see it was in the hinge region that kinds of turns the tip of the tail fiber so that the receptor binding protein of the tail fiber makes a better contact to the receptor of the bacterium. Now, as I say, there's no free lunch in nature, and everything comes with a trade-off. Like, for instance, mutations in the bacterial receptor that will have as trade-off reduced fitness or resensitization to antibiotics. Phage training can also have its trade-offs, and here you can see an example. So we had a patient with the Staphylococcus epidermidis infection. At that time, we didn't have any good Staph epidermidis phages, but we had a good Staph aureus phage, phage ISP, which we know has some activity against Staphylococcus epidermidis, as you can see in this picture on the right. ISP had some moderate activity against 3 out of 14 Staph strains from a collection, including the patient strain. You can see here in orange, moderate activity. Now, when this phage was pre-adapted or trained using 8 rounds of the Appelman's protocol, it was trained on the patient strain, we saw that it became really active against the patient strain, but we also saw that it lost host range. It was not active anymore against the 2 other strains from our collection. So often when we specialize a phage to become more active on a particular strain, it will lose activity on a broader set of bacteria. Some advice: use phages in synergy with antibiotics. We see phages as an additional tool. Select the adequate phages. So try to avoid using the same phage cocktail over and over again. I think only using the phages that are active is good for sustainability. So we really think that it will select for less phage resistance in bacterial populations. Of course, this is not always possible. So sometimes you don't have time to test, to look for the adequate phages. Then try also to pre-adapt phages when necessary and if time permits. And of course, you can develop some phage cocktails for first-line use where you don't have time to select the correct phages or to pre-adapt. So in summary, as Sabrina always says, phage can make antibiotics great again. Now, these cases I discussed were all salvage cases. And these are not what people call a controlled clinical trial. But our magistral phage preparations can also be used in controlled trials. And this is an example of an ongoing trial in the largest university hospital in Belgium. They published the study protocol, which is called Phage Force. And this is a controlled prospective observational study in which standard of care antibiotic treatment will be compared to standard of care antibiotic treatment plus personalized phage. It's not one product, one phage product. What is tested here is the concept of personalized phage therapy. This will be performed in a few indications that are mentioned there. But personalized phage therapy is also quite difficult logistically. What you can see here is the transfer of phage, bacteria, and sometimes patients in these 100 cases. So for each of these lines, you need to deal with a shipper, let's say DHL or FedEx. You need to deal with customs, because when these phages enter another country, there will be questions. What are these? Are these drugs? Oh, it's a virus. So I had, for instance, DHL calling me at night and asking me if this virus was not dangerous for their handlers. And then you also need to deal with regulators in all these countries. So it's what I've called my logistical hell, because this is for 100 patients. So that's why we focus our research now on developing what we have called a phage printer. And it would be a box like this. One would add the patient sample. And then this sample would be sequenced. AI would predict a phage genome. And then using synthetic biology methods, this phage would be produced. And we would have a personalized phage product. So this would mean instant point-of-care synthetic production of phages. This is a conceptual schematic I made of such a device. Now, let's see how it would work. First, you would have classical sample prep. So your sample would be homogenized, mixed with some buffer. And then this sample would enter the device. Now you see it will enter at two levels, at the bottom of this cartridge and at the top here, the top and here at the bottom. Now everything starts at the bottom here, where you will have bacterial cell lysis. So we need to lyse the bacteria. Then we need to extract the DNA and then sequence the DNA. And then based on the sequence of the bacterial pathogen, can also be more than one pathogen, using AI and the connection to an external database, a phage genome will be predicted. It can also be more than one phage genome, will be predicted to infect the bacterial pathogen that's in the sample. Then this genome can be synthesized, and then this synthesized genome can be transcribed and translated into phage using a cell-free phage production system. And then these synthetically produced phage can be purified. Then we end up with purified synthetic phage, but it's only a predicted phage. So we need to check if it's really active. And there you see that the sample with the living bacteria that was split two ways comes in handy here, and to really test the activity of the predicted phage. And then when this phage is active, it can be diluted in the excipient, and then we get these files with phage, personalized phage. Now this concept was published, I think, a few weeks ago, so you can look at that paper for more details. Now, when we do this today, this is all the equipment we need. So it will take like almost an entire lab of equipment. It will take very long time. It will take it will also ask for a lot of investments. For instance, to produce a synthetic phage genome today, it costs about 20,000 euro. But this is how the first computer looked like. And today we all have a smartphone that does a billion times more. It's small, and also, at least some of them, affordable. So this concept of synthetically produced phage, and also like a community-based system to source the phage genomes and also the bacterial genomes, because we need a lot of data to have AI predicting the correct phage. So for this, we thought of a community-based system, and yeah, an entire system. I will not go into detail, but you can read the paper. And we feel that this should be managed by an organization, for instance, by WHO. And of course, we would need to have some kind of phage exchange system. I think this is also important today, even without having a device like that. I think it's important today that we would have like an easy exchange of phages between labs and between countries. I would like to thank all my colleagues and also colleagues from the University of Leuven, with whom we collaborate for many, many years. And I would also like to thank you for your attention. So I'm open for questions.
So thank you, Jeffy, for such a wonderful talk. It's always a pleasure to have you. And thank you for, accepting invitation in such a short notice. Thank you.
You're
Thank you.
welcome.
And it's really fascinating the way you have portrayed the future of phage therapy, the personalized automated system. And as you rightly pointed out, yes, if we work together, this future is not much far away because we have, I think, all the parts of the system is available that is scattered. We just need to, make one system, which will not be a problem, right?
Yeah,
So,
exactly.
so I think there are no more questions. So once again, thank you.
I think they already asked their questions to Sabrina, I think. So-
Yes, there were so many questions were there regarding antibiotics and all. But I may tell the community that Professor PIRNAY is always forthcoming because we are also planning to have some regulation in India and reaching to ICMR and other bodies to, follow the same what they are doing, which kind of eliminating the requirement of GMP and other things. And it's
Yeah, I
work
did.
always
Of course.
rightly. He always says that you need now, you cannot wait till patient
Yeah.
die. Right. So for that also, thank you so much, Professor Pinay. And I hope you will guide us time and again to take this fast therapy in India very soon.
You are welcome, but I forgot to mention in the presentation that this system now a few months ago was also adopted in Portugal.
Yeah, yeah.
There's a few other countries that are thinking of applying this system.
No, this framework is really good. I think we can convince our regulator that yes, we can follow this. At least time being, then we can evolve with time. If there is a requirement, you can add some more checks and all. But this will definitely help us to push it through. So once again, thank you so much from the organizer and everyone from India. Thank you so much.
Welcome. Bye-bye.